Rahim. Once again, welcome to my lecture. So today we will talk about the antibody, that what are antibody, which is the end of chapter number 3. So in previous lecture we have discussed about the antigens, that substances capable of stimulating the formation of antibodies. So antibody can be defined as these are the special substances, these are the special proteins which is produced by the immune system that is capable of binding with high specificity to an antigens. As in the basic concepts of antigen, we have already discussed that antigen, there are two type of antigens. There are some antigens which are toxic, and there are some antigens which are beneficials. Those antigens which are present on the surface of red blood cell, so it come under the category of beneficials. They are beneficials, or you can easily find out the different type of blood group, while there are some antigens which are toxic, which produce toxin. These antigens, they are in the form of microorganisms, in the form of bacteria, in the form of virus, in the form of fungi, in the form of algae, in the form of many and more microorganisms which cause diseases in human bodies. So these antibodies which are proteinaceous in nature, so they are always produced against these particular specific antigens. As we know that antigens are typically other protein, but maybe carbohydrates, small molecules, or even nucleotides. Antibodies are powerful research tools because they bind specifically to a unique epitope. You know, epitope, these are the antigen determinant sites. Epitopes, these are the antigen determinant sites on the antigens, thereby allowing the detection of specific protein and an assay while avoiding the detection of unrelated protein. So it means that antibodies, these are the powerful research tools through which we can say that because of this binding property, that how it can binds with the epitope antigen determined site, thereby allowing to detect the specific protein which is present there while avoiding the detection of unrelated proteins. And this specific binding capability also allows antibodies to use in diagnostic application such as pregnancy test, and you can say in therapeutic application such as like chemotherapy, cancer treatment. So it means there's antibody, they are very important. So antibody play very key roles and the diagnostic procedures which we are performs in laboratory like pregnancy test, and so these antibody, they are also especially and therapeutic application, like let's suppose if you are going to treat a person who is suffering from cancer, so and cancer treatment and chemotherapy, so we are using different type of antibodies. Now I will talk about the classes of antibodies. So this is another word you can say an antibody is also known as immunoglobulin. The second name of antibody is also known as immunoglobulin, while antigens are also called immunogen. So it is a large Y-shaped protein. So look at here, it is produced by the B cells, lymphocytes, B cells, and used by the immune system to identify and neutralize the foreign objects such as bacteria and virus. Look at here, this is the shape, structure of antibodies, Y-shaped structure, Y-shaped proteins. This is what these are, antigen binding sites. These are antigen binding sites, and this is variable. This is variable portion, and this is constant portion. It can be changed, and this is constant. And there are two chain, one is light chain and one is heavy chain. Now I will discuss about the classes of antibodies. So these are the classes of antibodies: IgM, IgD, IgA, IgG, and IgE. So there are 5 different classes of antibodies. So there are different type of antibodies which are present in your serums and different concentrations. So look at this table. IgM, this antibody is present in your serums almost 5%, and IgD it is present in 0.3%, IgA is 7 to 15%, and IgG is 85%, and IgE is 0.02%. So it means that the highest concentrations of the antibody which is present in your serum, that is what? That is your IgG antibody. So it means that your serums it contain abundant concentration of IgG. Look at its structures. So the structure of IgM is monomer, pentamer, IgD is monomers, IgA is monomer dimers, and IgG is monomer, and IgE is monomer. Now what are the biological function of these antibodies? So it is also very important. IgM antibody, its biological function is this is the membrane bound immunoglobulin on the surface of mature and mature B cell lymphocytes. First, these antibody produce an primary response to an antigen. Let's suppose if the antigen, if it doesn't attack on your body, so it means that the antibodies become unable to produce. So these antibodies are actually produced against these antigens in order to induce the immunological reactions. First antibody produced by the fetus, efficient in binding antigen with many repeating epitopes, such as viruses, classification, and complement activation. While the IgD antibody, this is the class of antibody, you can say the membrane which is bounds immunoglobulin on the surface of mature B cells. There is no biological effector functions known still, because this is present in a very less concentration. Look at very low concentration, 0.3, and IgA also 0.02. IgA antibody, the biological function of Igg antibody, this is predominant antibody class and secretion, and this antibody is predominantly they are found in the saliva, tears, breast milk, and mucosa. So this is Igg antibody. This is the first line of defense against infection by microorganism. The IgG antibody, you can say IgG antibody is present here, most abundant class with 4 isotopes: IgG1, IgG2, IgG3, and IgG4. IgG antibody has the capability to cross the placenta, and they are also responsible to cause opsonization, and opsonization phenomena I will discuss it in my next lectures. While IgG antibody defends against parasitic infections, IgE antibody it is associated with hypersensitivity reaction or allergic reaction, which is found mainly in the tissue. So it means that IgE antibody, this is its biological function is that they are directly associated with hypersensitivity reaction like allergy, which are mainly found in our tissue, depends against parasitic infection. And this is the end of the chapter.